ischemic stroke and myocardial infarction, are two of the leading causes of mortality. Both conditions are caused by arterial occlusion, resulting in ischemic necrosis of the cells in the cortex and heart. Long non-coding RNAs are a group of non-coding RNAs longer than 200 nucleotides without protein coding potential. Thousands of long non-coding RNAs have been identified, but their involvement in ischemic stroke and myocardial infarction has not been studied extensively. Therefore, this study aimed to identify the role of long non-coding RNAs, particularly those that are commonly altered in these two ischemic injuries. We combined diverse RNA sequencing data obtained from public databases and performed extensive bioinformatics analyses to determine reliable long non-coding RNAs commonly identified from these datasets. Using sequence analysis, we also detected the long non-coding RNAs that may act as microRNA regulators. We found several altered long non-coding RNAs that were common in ischemic stroke and myocardial infarction models. Some of these long non-coding RNAs, including zinc finger NFX1 type containing 1 antisense RNA, 1, and small nucleolar RNA, host gene 1, were previously reported to be involved in the pathogenesis of each of these models. Interestingly, several long non-coding RNAs had binding sites for mRNAs that were previously reported to be involved in the hypoxic response, suggesting the possible role of these long non-coding RNAs as regulators in ischemic responses. The long non-coding RNAs identified in this study will be useful in determining the regulatory networks in ischemic stroke and myocardial infarction, and in identifying potential specific markers for each of these ischemic diseases.